Assalamualaikum. My name is Dr. Gabriele and I'm a lecturer from University Malaysia Sarawak, or in short, UNIMAS. And beside me is...
I am Teknowidi Anisah, a master's student from UNIMAS.
Today we are going to talk about the usage of molecular docking as a pre-lab screening tool for testing the effectiveness of phytochemicals from plant extracts.
What are phytochemicals and why are they of interest to us?
Phytochemicals are non-nutrient compounds that are produced by plants, fungi, or even secreted by animals in order to protect them from bacteria, fungus, and insects. These diverse types of compounds have been proven to have positive or medicinal benefits when ingested or even used as a lotion. They can act as antioxidants, anti-cancer, cholesterol-lowering, or anti-glycemic compounds. Indigenous people are very well versed in using specific plants or fungi as a source for treatment. Therefore, we have a rich choice of plants, respectively their phytochemicals, that can be used against specific medical conditions.
How do you isolate these phytochemicals? What do you mean by plant extract?
In order to isolate any of the phytochemical compounds, we need to extract them from leaves, stem, fruits, flowers, or even the root of the plant of interest. For the extract, we use various solvents. It can be water, methanol, or any other solvent. Once we have the extract, we freeze dry it, and then we have the crude extract. With that crude extract, we can start looking for the phytochemicals and their properties. Interestingly, plants contain thousands of different compounds like flavonoids, alkaloids, carotenoids, and so on. And they have numerous reports regarding these chemicals in reducing blood sugar or being antioxidant and anti-cancers. So how to find the appropriate phytochemical to answer our research question? Normally, one would do in vitro screening first and then use the most promising compound. But this takes time, a lot of time and money. But is there a way to shorten this process? Can we use an in silico method as pre-screening tool? For example, molecular docking. Tekno, can you explain exactly what is molecular docking?
Sure. This is an approach through software for us to see if there is any reaction between one compound with another. Of special interest are proteins, for example, receptors. This method requires molecular docking software such as Orthodoc-Vina and also Chimera. So how do researchers usually incorporate docking in their study? The initial step involves researchers choosing a specific plant and conducting tests on cells or animals. Subsequently, they delve into scientific literature to gain deeper insights into these compounds. In order to evaluate the potential of these compounds for treating diseases, they usually further analyze the compounds through molecular docking. This method examines whether the compounds can interact with disease-related proteins. If promising compounds are identified, further laboratory works can be conducted on cells or animals to comprehend their effects. We are determined to have a different approach from the usual. This new idea came out while wondering if there is a pre-lab screening method that could eliminate compounds that are not effective in vitro and in vivo studies. To approach this, we will start by screening through literature review for the plants reported phytochemicals. We take advantage of the abundance of protein and chemical compounds databases online. For example, you can simply access the compound databases in PubMed or PDB databank online. Then, we will run the in silico studies via Orthodoc-Vina and Discovery Studio and identify the compound with highest affinities. Through that result, we will run the cell or animal studies using the identified compounds in the laboratory and test their bioavailability. Let me give you an example. In our case, we are interested in phytochemicals that prevent the damage of bone cells by advanced glycation end products called AGE. From previous studies, we had already results from a plant called Tinospora crispa. This plant is found in Malaysia or Southeast Asia, and indigenous people use it, for example, to reduce high blood glucose levels, basically diabetes. Another plant from the same plant family is Tinospora cordifolia. This plant is used in Ayurveda against diabetes. Looking through the literature, we found that high glucose levels lead to a chemical process called non-enzymatic glycation of proteins, lipids, and nucleic acids. These so-called advanced glycation end products, or AGE, causing, for example, oxidative stress and damage to bone tissue. AGE have a specific receptor on most of the cells in the body, for example, bone cells. They bind to a receptor called the RAGE. To reduce the number of potential anti-RAGE candidates found in T. crispa and T. cordifolia extracts, we look for an in silico approach. So we use molecular docking to calculate the strength of a bond formed by a selected phytochemicals and the binding site of a protein, in this case RAGE. Next, we will be testing these compounds in the lab.
Okay, so now we have shown that molecular docking can be used as a pre-lab screening tool. Why do I say that? We have shown that plant extracts are having many phytochemicals, and it is much easier to do an in silico approach to select the most potent phytochemicals. So in our case, we were using T. crispa and T. cordifolia extracts and chose the most potent phytochemicals which will bind to our receptor of interest, which is the receptor of advanced glycation end products. Thank you.